Welcome to The Explainer. Today, we're diving into a genuinely fascinating medical mystery, one that honestly reads like a biological thriller. Imagine surviving one of the absolute most grueling medical procedures out there, a full heart transplant. You make it through the surgery, your new heart is beating perfectly, and you think, okay, I'm finally in the clear, right? Well, for one 74-year-old man, surviving that massive hurdle was actually just the beginning. Shortly after his transplant, his health started failing again, signaling that he was facing a deadly, completely unseen new threat. We're going to walk through the exact data and lab work his doctors used to crack this case. So just weeks after his successful transplant, this patient's blood work came back with a frankly terrifying number. His hemoglobin level had plummeted to a dangerously low 5.6 grams per deciliter. To put that into perspective, a normal range for an adult male is usually sitting somewhere between 13.5 and 17.5. So dropping all the way to 5.6, yeah, that set off immediate blaring alarm bells in the cardiac intensive care unit. His body was literally starving for oxygen. His doctors had to act fast, pumping in multiple units of transfused blood just to keep him stable, while they hunted for whatever was causing this. Part 1. A post-transplant mystery. The inciting incident.
Part 2. Decoding the blood work. Investigating the lab results.
The patient was readmitted to the hospital for a condition known as persistent pancytopenia. Now, if you aren't familiar with that term, it basically means his body was experiencing a severe drop across all three major blood cell lines. We're talking the red cells that carry oxygen, the white cells that fight infection, and the platelets that help your blood clot. When you see pancytopenia in a post-transplant patient, the differential diagnosis is incredibly broad. It could be medication toxicity, a raging infection, or an immune-mediated process. The doctors had to figure out exactly what was wiping out his blood supply.
When you really break down this critical data, something fascinating jumps out. While all of his blood counts were abnormally low, they weren't dropping at the exact same rate. His white cells and platelets were hovering just below normal, but the most drastic, life-threatening plunge was in the red blood cells. His anemia was wildly out of proportion to the other deficiencies. This was a brilliant observation because it allowed the medical team to narrow their focus. This wasn't just a general attack on his blood. This was pointing directly to a primary erythroid crisis. Basically, a highly targeted attack on his red blood cells.
Part 3. Narrowing the suspects. Finding the mechanism. To find the specific culprit, the doctors had to answer a fundamental question about how these red blood cells were disappearing. They had to figure out if the blood was actively being destroyed in the body or lost to bleeding, which we call hyperproliferative anemia, or if it simply wasn't being manufactured in the first place, which is hypoproliferative anemia. Think of it kind of like a car dealership running out of cars. You have to ask, is someone stealing the cars right off the lot, or did the factory suddenly shut down production? To answer that, they looked at his reticulocyte count, and it came back at less than 0.5%. Reticulocytes are those young, newly produced red blood cells. If a patient is bleeding or destroying red blood cells, a healthy bone marrow factory is going to work overtime to pump out new reticulocytes, meaning this number will be very high. But here, the reticulocyte count was profoundly low. So the absolute crucial takeaway here is that this proved beyond a shadow of a doubt that the bone marrow factory had just completely stopped working. This huge clue led the hematologists to suspect a really specific condition, acquired pure red cell aplasia, which is essentially a highly targeted strike on the red cell assembly line. The body just flat out stops making the precursors to red blood cells, while leaving the other cell lines mostly alone. But you have to ask, what could cause such a highly specific attack in a guy who just got a new heart? The doctors pulled together a lineup of the usual suspects. On one side, they had potential friendly fire from his own medical treatments. To keep his body from rejecting the new heart, he was on heavy immune-suppressing medications like mycophenolate mofetil and tacrolimus, and those are definitely known to sometimes suppress bone marrow. On the other side of the lineup, they had hidden viral assassins. Because his immune system was suppressed by those very medications, opportunistic viruses like Epstein-Barr, cytomegalovirus, or parvovirus B19 could be running rampant. Part 4. The bone marrow clue. Hunting for the smoking gun. Initially, the team carefully lowered the doses of his immunosuppressive meds, really hoping his blood counts would bounce back. But they didn't. His condition only got worse. They knew they needed a much closer look. A peripheral blood smear confirmed a total, absolute lack of new red blood cells, along with some abnormal variations in cell size and shape, which is a condition known as anisopoikilocytosis. Realizing the answer wasn't going to be floating around in the peripheral blood, the doctors authorized a direct investigation of the bone marrow factory itself. The team performed a bone marrow biopsy, literally taking a core sample from inside his bone. And wow, here is the smoking gun. Looking under the microscope, the pathologists found giant proerythroblasts, which are the very early stages of red blood cells. But they were swollen, and they contained eosinophilic intranuclear inclusions. Now, to a trained pathologist, these inclusions aren't just random. They are the unmistakable microscopic footprints of a very specific viral killer that had completely hijacked the cells. Part 5. Catching the viral culprit. Identifying the assassin. The microscopic evidence pointed directly to a highly specific and honestly surprising culprit, parvovirus B19. This is a single-stranded DNA virus that is notoriously specialized. It doesn't just attack the body randomly, no. It preferentially targets, enters, and infects erythroblasts, the exact cells in the bone marrow responsible for making red blood cells. It binds to a specific receptor on these cells, sneaks inside, and completely shuts down the production line just so it can replicate itself. As Dr. Charles S. Azar, one of the physicians on the case, noted, the findings suggestive of parvovirus B19 infection from the bone marrow biopsy were unexpected. However, in hindsight, they accounted for this patient's presentation. It perfectly, exactly explained his persistent anemia, his massive bone marrow suppression, and his complete lack of reticulocytes. To confirm just how bad this infection really was, they ran a quantitative DNA test on his blood, and the results were literally staggering. He had a massive viral load exceeding 100 million copies of the virus per milliliter of blood. And get this, the testing assay actually maxed out. It literally couldn't count any higher than 100 million. Because his immune system was chemically suppressed to protect his new heart, his body couldn't mount a normal antibody response to fight the virus, which just allowed it to replicate to astronomical levels. Part 6. Resolution and takeaways. Curing the unseen threat. So the ultimate question for the medical team became this crazy balancing act. How do you fight off a rampant, out-of-control virus when a patient's immune system must remain deliberately suppressed to protect their new transplanted heart? If you lower the transplant meds too much, he loses the heart. If you don't fight the virus, he loses his blood. The medical team deployed a careful, highly balanced three-step counterattack. First, they cautiously reduced his immunosuppressive medications, just enough to give his immune system a fighting chance. Second, and most importantly, they administered intravenous immune globulin, or IVIG. This is essentially borrowing antibodies from healthy blood donors to do the fighting for him, which dramatically knocked down that viral load. And third, they provided supportive care, with continuous red cell transfusions to keep him alive until his bone marrow factory could safely reopen. Following the IVG treatments, his viral load plummeted, his bone marrow started production, and his anemia finally resolved. But what's truly fascinating to take away from this explainer is the actual identity of the culprit itself. Parvovirus B19 is incredibly common. It's a usually harmless childhood bug. It's the virus responsible for fifth disease, which is that mild illness that causes a slap cheek rash in kids. By adulthood, roughly half the population has had it and carries antibodies against it, literally never giving it a second thought. But in the unique, immune-suppressed environment of an organ transplant patient, this harmless bug turned into an opportunistic master assassin. This whole explainer just brilliantly illustrates the delicate tightrope walk of transplant medicine and how a suppressed immune system can turn a distant childhood memory into a life-threatening crisis. It really leaves you wondering, doesn't it? As medical science allows us to manipulate the immune system to perform absolute miracles like heart transplants, what other seemingly harmless microbes are just waiting in the wings for our defenses to drop? Thank you for joining me on this incredible case, and keep questioning the science around you.